Well, good morning, everyone. Can you hear me? Welcome to the annual SWOG Clinical Trials Partnerships Update Forum, which we are doing at the fall meeting until we decide to change it back to every 6 months, but this seemed to work good this time. And we are talking about behind the scenes and what's gone on, as well as on stage. What 3 protocols in particular are maturing, and you'll hear about them from their investigators this morning. I'm Kathy Albane, medical oncologist at Loyola, to the west of where we are in Maywood, Illinois. And with me on the stage are Ms. Casey Dawson and Ms. Crystal Miwa, who are very important part of what I call the 3 C's. The third C, will you stand up so they can see you? Chrissy Laubach. So operations and group chair's office represented by this group. Also acknowledge Dr. Well, let me move to the next slide. I want to acknowledge everybody, and I'm not going to read all these names, who have been really pivotal in getting us to where we are right now with clinical trials partnerships, and I will say CTP from now on, so remember clinical trials partnerships. The executive committee, the committee chairs and executive officers, a scientific advisory board, which will be meeting in closed session tomorrow, advising us experts from across all disease sites, as well as the executive committee. Then the CTP Protocol Operations Office, directed by Dana Sparks. Dana, are you in the audience by chance? And her deputy is up here up front, Chrissy Laubach, and most recently the addition of Katerina Gasik. Katerina, are you here? Thank you very much for your important addition to our group. And administrative assistants, my right hand, Julianne Russo. Julianne was waved to the group in my office. And then Whitney Leslie, before Julianne was hired, was very important. And Valerie Derling from the group chair's office, and our attorneys and budget analysts. So this is our agenda for this morning. I'll first remind you of what the Preferred Partnerships Program, or PPP, is within clinical trials partnerships, then move on to discuss the development process. So those of you that are on committees not yet involved can see what's involved from the start. And I'll share this throughout the presentation with Crystal, who will take you, I'll start the early part, and Crystal will take you through to protocol activation. We'll hear from our 3 study chairs on projects in leukemia, head and neck cancer, and muscle invasive bladder cancer, nearing their final contracting phase. They're all approved already by the executive committee. And then some other studies under discussion, and one other model we have actually accruing right now. And last but not least, what SWOG sites can expect as these trials now move through the process and protocol activation. And Casey Dawson and Crystal Miwa will give that presentation at the end. So SWOG CTP establishes and administers scientific preferred partnerships between SWOG and industry, fully funded by industry. It's a mechanism whereby our large network of institutions collaborate concurrently with study design, development, all the way to activation and conduct of the study with the industry partner. So this differs from the IIT type of trial you're used to, where industry provides some components of the funding, but not the entire package. The SWOG PPP, the preferred partnership, offers a range of study platform options from phase 1B through potentially a registration model. It facilitates testing of novel agents in combinations or across disease sites, within a disease sites, or, as you'll hear in leukemia, an uncommon scenario where it's very difficult to accrue patients, and thus needing special help and special funding to do the trial. These trials flow through dedicated CTP channels. A lot will be, most will be very familiar to you because utilizes the same operations, statistics, data management personnel, funded separately, not through the NCCTN mechanism. And so this schematic summarizes what I've just said. Let's see here if I can point, yeah. So a CTP study can be also federally funded. That's not under my purview, and we won't be discussing these. But as you know, multitudes of SWOG studies over the years have used industry collaboration, funding, drug costs, very successful model in our group for many years. But over here, these are the fully industry-funded trials that are designed together from the beginning. So it can be, as you'll hear in the head and neck trial, a single committee-initiated study. We used to call these clinical trial initiative trials. These aren't like an IIT at your institution where you propose it and get some funding. This is a joint development with the company. And we hope if successful, these can be broadened into more collaborations with the same company and perhaps even move to a PPP, which is the mechanism I just reviewed, which takes the pipeline or complex multi-arm platforms, as you'll hear in the GU study, joint scientific development and governance, and a dedicated CTP infrastructure. Now, all of these then, all fully industry-funded CTP trials cannot overlap or compete with any active federally funded trials in our group or others that are known to be about to open in a sort of the old intergroup type collaboration. So, the process. Again, joint study design, scientifically, budget and contracting, and protocol development and study build. I do want to just shout out to Mike LeBlanc. Mike, you want to wave to everyone? I think everyone knows you. Oh, you moved to the back. Here I thought you were going to break stride. Anyway, Mike and his group are extremely critical to the novelties of the success of the novelties of these trials, and we're learning on some of them as we go and looking forward to continued collaboration. Now, not going to read all these boxes per se, but let me just introduce you to this diagram. It's extremely important. The red across is the company. The purple ones are scientific. Blue is budget. Green is contracting, and then study build, forms design and development down here in yellow. So I get this question asked at every meeting and very frequently in between meetings from the various committees. Well, what do we have to do? We're talking to a company. What do we do first? We have an idea. So the very first thing is that the SWOG committee, the committee chair or a collaboration of chairs sometimes, agrees on the idea, basically the basic elements of study design. And they then go to what's called the feasibility step. And what's involved there is about a 3 to 4 page form is filled out by the committee. This is not a capsule, this is before that. It's just, I'm not gonna read all of these, but basically, as you can see here on these 2 slides, just has the basic components. So the SWOG Executive Committee can then review it and decide, yes, it's feasible to start the process, meaning yes, we will dedicate personnel and operations in contracting and budgeting in the statistical center and data management areas. So that has to be approved, and then if it is, hopefully it's a go and not a no-go. We actually have one in the GI committee I'll mention later that's just at this step. So then next, it goes to the executive, now it doesn't quite go to the executive committee. There's a statistical part here. There is a budget that's worked up that the company agrees. And once all of these are in order, then it goes to the executive review committee for approval of the capsule. So this is a typical capsule. The company will have also read the capsule and inputted on it. Same kind of capsule model that you're used to in the disease committees. So the Executive Review Committee for CTP replaces the NCTN Steering Committee reviews of the initial capsule and then later for the full protocol. There's a core group that's selected from the Executive Committee, the Scientific Advisory Board, and 2 ad hoc reviewers with special project-specific expertise. It's a rigorous review, not a rubber stamp. The capsule template is modeled after the NCTN version, very similar, and a standardized voting process that's also very similar, with an open session presentation by the study with the lay advocate who's been involved in this development, and then a closed session and a vote. So with that, I will turn it over to Crystal Mimo. You want to join me up here? And whoops, oh, no. All right. There's your pointer. Where is it? There it is. It's there. See it?
All right, so as Dr. Albane said, we do a lot of different processes right before we get that final approval to move forward. And as part of that, we are working very closely with our budgets and contracts group to identify all of the tests and procedures that will be performed on this study. This helps with all of the budgets to identify what needs to be in the contract so we can get that final contract executed with our pharma partners. And that's really kind of our go point. Once we get that contract signed, we get into protocol development. The protocol development mirrors a lot of what we do on the federal side, at least on the steps that we do. And so kind of mirroring what that is, we have our 4 protocol drafts that are created. The first one's usually got a lot of holes in it. We're still looking for some additional information to include in that. We still are going to be doing the rapid. If you all sat in on rapid, it's the wonderful 4-hour phone call where we go page by page in the protocol document, identifying any gaps of information that we still need to consider. It will go through the stats PRC review as well. And then finally, it will be, there's a couple parallel steps that will happen, including the FDA submission, a submission to the CTP Executive Review Committee, which is very similar to the CTEP process review. And so there will be those final checks and balances that we will do before going to the WCG IRB and finally getting it activated to these sites. So a lot of different things do happen while that protocol development is happening and we're writing the protocol. And that also includes all of the form builds and the nebula that we will be using as part of this build as well.
Great, thank you. So all 3 of the studies that you're about to hear from are really right here, floating around here. So we're very hopeful that in the next 6 months we will be in full steam, but protocol development's already starting. We're in the final contracting and budgeting for the most part, but the companies have approved going forward with the budget that we submitted. So that's all good. So we have then active industry collaborations in acute lymphocytic leukemia, breast cancer triple negative disease in the adjuvant setting, metastatic setting, ER positive, HER2 negative, bladder cancer, and head and neck squamous. And then pending collaborations, and a lot of these will be discussed in the Scientific Advisory Board. We do have one that is very close, as I said, at the go, no-go step. The form is actually imminently due to go to the EC, so that's good news. So we'll have more news about that. Several company pipelines, several that we're interested and have we've been put on hold a lot because of changes in the company, changes in the leadership of the company, sometimes changing agents. So we still are keeping a list of those that are potentially interested as we move forward. And also the SWOG VA committee is an ideal place to think about some unique collaborations, and we haven't matured that yet. So we have one PPP, or preferred partnership, and that's with Novartis. That master agreement's been signed. They have a pipeline that they're interested in having SWOG help develop. And this primarily is in solid tumors, hematology, immunotherapeutics, and early therapeutics in rare cancers. We anticipate a call with the senior leadership later this fall. This one has been put on hold for a while, this call, because of change, a lot of change at the senior level in the company. So we have one person who's been with us from the get-go, and she is shepherding this through the new group right now. But we thank Novartis for being with us, and we're looking forward to expanding. But we do have one study. And I believe it's a good time for you to unmute yourself if you're on. Let me see, the leukemia trial, yes. Are you on, Dr. Lieke? Michaela Lieke?
Yes, I'm right here. Can you hear me?
Can everyone hear? Good, okay, go ahead with your first slide and tell me when you want me to advance to your next one.
Okay, perfect. Well, thank you for giving me the opportunity to present our study here. And I also want to thank the leukemia chairs, Dr. Erber and Dr. Advani, for letting me work on the study. Thank you to our wonderful statistician, Megan Otis, and Chrissy Laubach. The study wouldn't have come to fruition without you, so thank you very much. Our study is a phase 2 trial of asciminib, dasatinib, prednisone, and blinatumomab in patients with newly diagnosed Ph-positive acute lymphoblastic leukemia. And just to give you a little bit of background, Philadelphia chromosome-positive ALL is caused by the BCR-ABL tyrosine kinase fusion protein, and it is actually quite common in older adults. In younger patients, combination chemotherapy that's quite aggressive and includes a tyrosine kinase inhibitor, and oftentimes stem cell transplant is used. But that approach is too aggressive for most patients with this disease who are elderly. Therefore, to our older patient population, we usually give the tyrosine kinase inhibitor dasatinib in combination with a steroid like prednisone for induction therapy. This is associated with high response rates, but unfortunately, relapse is fairly common. And this is what our trial is trying to address. We would like to improve the response rates and reduce the relapse rates by combining dasatinib and prednisone with 2 other drugs. One drug is asciminib, which is another inhibitor of BCR-ABL, but it inhibits it by a different mechanism than dasatinib. This is an oral drug that's already been studied in combination with dasatinib, and a recommended phase 2 dose has been identified. And then post-induction, we want to include the bispecific T-cell engager blinatumomab that has a really good track record of eradicating minimal residual disease. So our study objectives are to study the rate and depth of remission of this drug regimen. We're also interested in the disease-free and overall survival. And lastly, there are correlative studies performed by Dr. Jerry Radic, who look at the mechanisms of resistance. From a statistical perspective, our study is a phase 2 trial. It's built on the hypothesis that the day 84 major molecular response rate will be improved to 70% with this combination compared to the reported rate of 52%. And we will need about 35 patients to be enrolled over 2 years to prove this hypothesis. Next slide, please. This is the study schema. I admit that it's probably a little bit dizzying, but we'll work through it. So on the focus initially on the left side. So patients will be registered to the study and will start their induction treatment with asciminib, dasatinib, and prednisone. After 28 days, they will undergo a bone marrow biopsy, where the expectation is that most of the patients will be in remission. At that point, they will complete an 84-day cycle, and at that time, then undergo another bone marrow biopsy, which is the biopsy that assesses our primary endpoint. If they're still in remission at that point, then they will be registered to post-remission therapy, where they receive 3 cycles of blinatumomab and dasatinib to clear all minimal residual disease, and then they move on to another bone marrow biopsy, and if remission is confirmed again, then they move on to the registration step number 3 and start their maintenance therapy, which lasts for up to 5 years. Going back to the top, for patients who on day 28 have not achieved a complete remission, they will undergo another bone marrow biopsy at day 56. The majority of patients is expected to achieve a remission by day 56, and they will then join the other group in post-remission and maintenance therapy. For the rare patient who has not achieved a complete remission, even by day 84 or day 56, they will then have the opportunity to undergo reinduction with blinatumomab for up to 2 cycles. If they don't have achieved a remission at that point, then patient would come off protocol therapy. We're currently in the process of writing the protocol, and we're hopeful that we'll be able to start enrolling patients at the beginning of the new year. Thank you.
Kayla Leakey, Dr. Leakey, for your presentation, all your hard work together with your co-chair. Thank you for putting these slides together and participating.
My pleasure.
All right,
Thank
I'd
you.
like to invite Dr. Josh Meeks, and in absentia in Turkey, I think, if he's there, Dr. Lerner, to come to the podium and discuss a very exciting platform trial that we hope is just the start of many more arms to come down the road. This is forward and back.
Great.
And this mouse arrow only.
Okay, thank you. Thank you again. So my name is Josh Meeks. I'm from Northwestern and a urologist just uptown here. As you can see from our list of investigators, this is a huge tent of people involved in this trial. And really wanted to thank Genentech, which is our clinical trials partner for this project. So the main question that we are interested in is there a better, more precise systemic therapy for patients with muscle-invasive bladder cancer? Really, SWOG set the standard for what is systemic therapy and providing neoadjuvant chemotherapy for patients. They usually get 3 or 4 cycles prior to going on to radical cystectomy. But there are still major challenges to this. About 40% of patients can get the therapy, so over half simply aren't platinum eligible. And then those who get it, only about 1/3 have a response. So there's 2/3 of patients that just have toxicity and really don't benefit from the therapy. Now, there's more precise targets that have been shown in other therapies, but unfortunately, we know with bladder cancer, there are just too few drivers. There's just too few oncogenic mutations that we can target. So instead of that, what we've tried to do to approach this is actually to use an RNA-based profile. Because every tumor has a profile, the thought is, can you leverage that to target the therapy to better individualize therapy? And again, many of the investigators from our leadership have been involved in that work through the TCGA. This is the schema for our trial, and it's a relatively big trial of around 400 patients, and I'll just try and walk you through this. So patients that are initially diagnosed with muscle-invasive bladder cancer, they have their tumor, an FFP specimen, sent off to Tempus, which is our companion diagnostic partner. And we've had to go through developing an IDE for this new biomarker. At Tempus, tumors are classified and given a subtype. And so this is pretty rigorous classification that's either a luminal tumor, a basal tumor, or neither. And then those results are sent back to SWOG and the site. Now, the patients are randomized 3-to-1 to a subtype-directed therapy versus standard of care, which again is neoadjuvant chemotherapy. So if they're randomized to a subtype-directed therapy, they'll get a target, which we hypothesize will be directed right at that specific RNA-based profile. So for example, in luminal tumors, which we've seen very high levels of FGF receptor 3, they'll receive an FGF receptor 3 inhibitor plus atezolizumab. In tumors that are more basal, which tend to have more immune infiltration, they'll get both atezolizumab and tiragolumab. And this is the first time that combination has really been used in bladder cancer. And finally, for patients that really don't fit into either luminal or basal groups, they'll get both chemotherapy as well as atezolizumab. After treatment is done, they'll go on to undergo a radical cystectomy, and the primary outcome of this trial is pathologic response. So we'll know very early if the therapy is working. The overall objective is to get a 15% improvement over the standard of care, so that's our primary outcome. We're hoping to open in, I believe, 2024, is that right? Yeah, early 24. So a few more checks to go along the way, but we're working in the protocol right now. Yeah, so again, a lot of this is sort of working through the protocol with Genentech, or like on our own to develop it. But then on the other side of it, because this is a brand new biomarker, it's interesting. So the work originally came with the TCGA. Our group worked to develop this classifier. So it's a single patient classifier where any tumor can be placed into that. That then had to go to Tempus, who doesn't normally do this kind of work. They do the RNA-seq, but not the classifier. So right now they're going through, they're sort of validating that work. And then once that's done, it'll go through the FDA and be approved with an IDE before going into the trial. And again, it'll be ready to go. Okay, thank you.
We have about 20-some participants on virtual, and so the chat's open if you have any questions. And those of you in the audience, think about questions, because we'll have plenty of time for discussion. Last but not least of the triumvirate here, with protocols close to getting ready to take off in the second half of our process diagram. Welcome Dr. Paul Swasicki, successfully commuted from Ann Arbor this morning, and glad you made it. Didn't get bogged down around the bend there. Welcome, Dr. Swasicki. That's what you're up to.
So first off, amazing trials that were presented right before mine, so I feel kind of bad with the simplicity of this one. Next, I'm so indebted to many, many people here, and I think Megan, we'll hear her name here, Megan Othis has been instrumental in designing this trial, but also my co-chairs, Dr. Tina Rodriguez out of Fred Hutch, as well as my colleague Chad Brenner, translational PI out of University of Michigan as well. And this is a study of carboplatin, paclitaxel, and can't name the drug because of contracting, but it's a bispecific EGFR antibody in metastatic head and neck cancer. I'm the co-chair of both of the experimental therapeutics group at University of Michigan. I specialize in head and neck cancer and rare cancers, and I realize that not many people know head and neck cancer. But in brief, for current metastatic head and neck cancer, although immunotherapy is approved, still has horrible outcomes. Response rate to immunotherapy, although it's changed the game, is approximately 13 to 14%. And now, even in phase 3 trials, our survival is about 13 months. There's a strong rationale currently for targeting EGFR as well as MET. EGFR is an established target. In fact, cetuximab has now been approved for more than 15 years. However, cetuximab, when approved, only increased the survival by about 1.5 months, technically 7 weeks, yet still got FDA approval. c-MET expression has been shown to be a critical target in this disease, including a mechanism of resistance to EGFR inhibitors and overall a prognostic biomarker for for tumors that are non-responsive to treatment. We actually don't know the best treatment post-IO. This is evolving very quickly, and I think medical oncologists in the room know that we can be somewhat scatterbrained at times, but a whole host of things are being explored in this region, and we know that chemotherapy is probably standard of care with something, we don't know what. Based on previous first-line data, chemotherapy of carboplatin plus 5-FU plus cetuximab is superior survival versus chemo doublet. However, as we can see here, the toxicities of head and neck cancer, what we expect as a standard of care, are pretty dismal. Grade 4 toxicities in about a third of patients, grade 3 or 4 in 82%. Platinum docetaxel and cetuximab is a much more tolerable regimen, although not category 1 for NCCN. This study drug is a bispecific EGFR and MET inhibitor. What's interesting is I can say it's approved in other diseases, and there's a lot of preclinical data showing that it has a lot more cytotoxicity compared to cetuximab. And interestingly, for a long time, head and neck cancer, we actually don't think cetuximab works through EGFR inhibition. We actually think it's a poor man's immunotherapy that's probably causing ADCC, and it's been shown that this drug is even more immunomodulatory than cetuximab. So there's a lot of preclinical support to explore this drug in mucosal head and neck cancer. And as aforementioned, MET overexpression has been shown to be predictive of a response with this drug in EGFR wild-type patients, not just those with mutations. The primary objective of this study is to establish the RP2D of the study drug plus carboplatin and paclitaxel and recurrent metastatic head and neck cancer, and establish the overall response rate of the combination in platinum-naive patients previously treated with a PD-1 inhibitor. This is a brief schema. It's a phase 1 dose finding followed by phase 2 dose expansion. Once we hit the RP2D, treatment is carboplatin and paclitaxel per standard of care plus the study drug. And then initially there's a conservative dose escalation strategy. Of note, the study drug has been explored in chemo doublets and even triplets. And I understand there's now quad to 5 drug regimens with this drug showing tolerable toxicities. And so we anticipate that this will be tolerated well. Eligibility include metastatic mucosal head and neck cancers of the wet lip, oral cavity, oropharynx, larynx, nasopharynx, and sinus. We're aiming to be as inclusive as possible to engage as many partner sites. We're trying to make as few restrictions as can be, ECOG 0 to 1, up to 2 prior lines in the metastatic setting, and must have received a previous PD-1 inhibitor. Patients are excluded if they were treated with a platinum agent in a metastatic setting, or they had received cetuximab for obvious reasons. Hopefully, we will keep on making progress and advance this therapy in the area. This is the first time this drug is going to be used in metastatic head and neck cancer. And so, yeah, look forward to advancing this to the clinic. Thank you.
I say a word how initially it was or how the process did work, the early part of our process, even during design?
Yeah.
Say it doesn't know how
I didn't know what you wanted me to include.
okay.
Yeah.
No, I just came
yeah.
to it just now.
Yeah, I think this has gone many changes in the design over time and is a testament to how CTV is flexible. Initially, this started out as a platform trial with 6 different arms, and actually the sponsor was extremely supportive and approved that, recognizing that there hasn't been a true head and neck cancer trial in SWOG actually since Dr. Baker was the head of SWOG. I forget how many years ago now, because he even retired from University of Michigan. We decided to go more conservatively, and SWOG was extremely supportive of a head and neck cancer trial here, but we scaled it back with the plans to possibly augment and build upon this if positive and if it accrues well. I will point out that IMET S2101 includes head and neck cancer and is actually accruing quite well in that disease type, so I'm very positive this will go well, just promoting another study I'm involved with. And so I think overall it's been a good experience, an amazing teamwork. I can't be thankful enough to the office and kind of meeting in the middle. And once again, STATS deserves a huge shout out for repeatedly redesigning this trial to support it.
Well, thank you very much, and hopefully there'll be some questions during the discussion period for the investigators. So the next trial is not quite as far along right now. It's a collaboration among the SWOG Breast Committee, AstraZeneca, and happily I can announce a full partnership with Guardant for their assay, ctDNA assay on this trial. So that's progress since the last meeting. It's called the MONITOR trial for metastatic ER-positive HER2-negative breast cancer on first-line standard of care aromatase inhibitor plus CDK4/6 inhibitor. This is chaired by Dr. Kevin Kalinsky. Project design is just went through another redesign to focus on the control arm and to help the sample size. So, but it's based on ctDNA status in general. If it's rising during this standard of care therapy, patients are randomized to multiple parallel arms with investigational agent or combinations of agents. And the budget is currently under active negotiation. So we hope to get it to the ERC soon. This slide just talks about some of our pending or potential collaborations. You'll hear more about this at a subsequent meeting, not this meeting. It's still being discussed. An academic drug development new platform for Roche Genentech, and we're actively discussing whether SWOG CTP would fit into this for basically global registration trials. There's a CNS working group that we hope to collaborate with here in SWOG. We've already started to talk to digital engagement and then put some, with the change in leadership there, we're on a pause, but hope to reinvigorate that, as well as I said earlier, the VA committees. The Immunotherapeutics Committee, we did have a possible idea and we went pretty far along with it, but it just really didn't have a good focus, so we elected not to pursue that particular industry collaboration. We do have this one with metastatic colorectal cancer to liver that will be involving the GI committee as well as immunotherapeutics and the surgeons. And then earlier in discussion, an anal cancer multidisciplinary trial. And the several company pipelines I alluded to earlier, and there are other committee chairs that have expressed interest, and usually these come up during the meeting or after, and the Scientific Advisory Board will be considering some of them tomorrow. This is, I mentioned at the beginning, we also have a different model. This is not a classic CTP trial. This trial is being run, it's a registration trial by AstraZeneca and Daiichi. The trial is open, but SWOG CTP has been the lead academic partner. On the left here, you can see pointing with the advancing, that's not gonna work. So these familiar names in the SWOG Breast Committee are the core steering committee. Dr. Bardia is the US overall and the overall chair of the trial. And then Dr. Nadia Harbach is for the non-US sites. We have a number of SWOG sites that are participating and going through the contracting and startup phase now. The trial is accruing, and we have a lot of excitement about this. If you want to hear more, there will be a formal presentation during the breast committee meeting. I believe that's on Friday, the full committee of the breast committee. Basically, we're looking at datopotamab deruxtecan with durvalumab or without it versus investigator's choice, and the option of continuing if they've had neoadjuvant. They can have neoadjuvant immunotherapy or may not have had. So the eligibility are as broad as it can be so that we can capture these patients who have residual disease with triple negative cancer after their neoadjuvant therapy. It's a very exciting trial. So we've been through a lot of the science of the committee and how studies are developing, but there's been a whole lot of progress backstage to fit in our theme with our theme today. So I'll invite both Casey Dawson and Crystal Miwa up to the podium to share this next part with you.
Thank you. And thanks to you all for being here. As Dr. Albane mentioned, a ton of work happening behind the scenes, and we owe so many people so many thanks. So I'm going to talk about, Crystal and I will talk about what sites can expect in our preferred partnerships and industry-supported trials. We'll touch on study feasibility and the site selection process, study funding, as well as the various systems that we'll be using for SWOG CTP trials. So getting started, member sites will receive a notification via email about studies that are available for CTP. So if there are sites in the audience, make sure that your contact information is up to date with our membership department. In that email, there will be links for the study synopsis, as well as the feasibility questionnaire and some information about study funding. We would ask interested sites to complete the feasibility questionnaire, and then there's an internal site selection process. And once a site's selected, we will complete a contract specific to that study with each participating site. Let me know if you want to add anything. And you as well. You're always welcome. So we won't be negotiating study-specific, site-specific budgets for these studies. CTP study funding will be set based on complexity of the study. So we'll look at what activities need to happen at the site and consider that effort. We will include, here's an example here of some cost categories. We plan on including site startup activities, per patient enrollment, of course, and then additional funding available available for study-specific requirements, non-standard of care testing, specimen and imaging submission. And I've included examples of some funding that's been recently approved at closer to industry levels for some upcoming CTP trials. I wanted to note that we don't have any invoicing requirements, so just like on our regular SWOG studies, payments will be triggered by milestones entered into the EDC, which is a nice segue into the systems slide. So first and foremost, I owe many, many thanks to the implementation team, as well as our colleagues at Crab and Hutch for all of the work that's gone into getting us ready to activate studies when the time is right, and that's soon. So we're going to be using the Florence system as our electronic trial master file. So we'll use the system for all regulatory requirements and communication with sites around those regulatory requirements. We'll have the investigator site file available for sites to use as they wish. Sites will be given access as soon as they complete the site study-specific contract. We're going to be using the WCG IRB as our central IRB and have negotiated funding for the coverage of those costs. Submissions will go through Connexus. I don't know if you want to add anything here. Site submissions through Connexus. There'll be study-specific training for sites. And then Crab Nebula will be our electronic data capture system, and we intend for it to look a lot like what sites already know and love. Again, thanks to our colleagues at Crab. And this is a nice slide that will just show what functionality Nebula will have. So instead of open and rave and CTEP errors, we'll be using the Nebula system for that functionality. We will continue to use the SWOG specimen tracking system as well as the expectation and query reports system, so no change there. Again, many, many thanks to everybody that's been involved in getting us to this point. I feel really good about all the work that's happened, and we are ready for action as soon as we get some of these study contracts executed. Would you like to add anything?
I think you did it perfectly.
Excellent. That's the end of my slides.
Oh, okay.
Sorry, I did think of one more thing. Sorry. Sorry about that. I will be available for questions always. You're welcome to contact me. Next meeting, at the spring meeting, we plan on having a table specific to CTP at the ORP Open Forum, which is very site-centered. So look for us there if you're a site and you would like more information. Thanks.
Well, thank you both for putting that together. So CTP milestones. I think you can see that we've had successful design and implementation of our internal processes. There's been upfront engagement among a lot of committee chairs, the advisory board, industry partners, and CTP leadership. Our process timeline, our process and our timeline for such, I will say here that we are going to put in tracking metric system for these trials as well, right, Crystal? And that's being worked on actively right now. So the front end, the first half of that process diagram is indeed, you know, some of those are like several years already. And you can tell it has to do with companies pulling drugs. This next two of our studies got, you may remember there was a lung trial, platform, multi-arm, that went back and forth to the ERC, was redesigned, and got pulled at the 11th hour from further development because of new data that came out on a competitor drug that changed the standard of care and on and on. And so it's just, you know, we had even got biodesics involved. It was just a very complex trial design. So these things happen, whether it's the CTP or whether it's on the NCI side. The other one was withdrawn earlier, the MDS trial, because of, again, new data and lack of encouraging results in the pilot studies that were done in other sites. These studies have many early and mid-career investigator opportunities. We actually have mid-career investigators chairing, as you heard from today. And on the, I don't know if Josh is still here, the bladder study, they've added mid-career investigators for each of the subtype arms to work under Dr. Flagg, who is the overall medical oncology chair of that trial. So there's exciting new initiatives to explore, pending with other companies, and more interested partners are welcome. So we have an email address that's monitored regularly by Julianne Russo, and we also have our website that you can go to, and feel free to contact us at any time. So your yellow light didn't come on. It's probably just about to over there. So we're here at the discussion period. And please go to Mike's. We have plenty of time to answer any of your questions. Are there any questions coming through on the chat? Now, I know the meeting's just starting, but we need to rev up here. Can I just ask, how many of you are from industry in the audience? Just raise your hand. Well, welcome to this. This is a good forum for you to attend. And thank those of you that are on any of these studies already for your support. How many site investigators, physicians are in the audience? So one of the things, I'll just interject here, that we struggle with is where to put this in the course of the group meeting. And as you know, the group meeting being so condensed that all the main committees are operating on Thursday and Friday with 2 plenary sessions. And, you know, if you put it this early, a lot of these folks that are coming to the full committees are just flying in or driving in today. So I'm glad to have the attendance we do have. Let's see the rest. Data managers, research nurses. There's a whole bunch of other people. Work at Crab or the Hutch. Any questions? Do we want to ask the audience any questions? Let's see. I know at our last open forum, there was a lot of concern from the sites about the payment schedule, and we've obviously, as they've discussed, now come a lot further along with the fixed cost that is competitive, although I will state that, you know, these registration trials that are run by the companies are much higher in per case payment, but there's no way we can do a 1,000-patient trial and have it be funded. I think for those of you with the industry partners, you know, remember what you're buying for your buck here is not just providing drug, providing site payments. You're providing for all this infrastructure that you normally do. You normally have your CRO, you have an auditing group, you have all that you fund. We're doing that. We're doing the data management, data collection. So it really is a great buy, so to speak, even though the costs seem high when you look at these budgets. Would you add to that, Casey, anything? You agree? Disagree?
Nothing to add.
Nothing to add.
They're also buying the SWOG network.
Could you use the mic, please? I'm sorry.
Thanks, Danny. I was just going to mention that they are also buying the SWOG site network.
Yes.
Which is a good bang for your buck.
And I mean, that cost would be billions of dollars, right, in worth that you're getting with these trials, our 1,000 plus number sites, yes. Well, yes, Dr. LeBlanc, thank you.
Following up on what Danny was saying too, compared to some industry funding, this might seem low, but it's high relative to NCTN funding. Understand you get extra credit for that. But the other thing, it's not going to be as much data collected as most industry studies. We're going to try to adhere the best we can to SWOG, NCTN standards, and then some to make that data collection efficient. So that's that really makes that trade-off, I think, much better in terms of the per case reimbursement. And the startup cost funding is substantial.
Any other questions? Well, we'll stay up here at the front if you want to ask them privately, feel free. Thank you all for attending. Have a great meeting. And see you around the halls of the Hyatt. Thank you.